if you take a look at this, primary level of structure is simply the number and pattern of amino acids, okay? And in this particular model, each of these little beads represents an amino acid, okay? So primary level of structure, the number and pattern of amino acids that make up that protein. What determines that? What determines what amino acids make up a particular protein? Very good, DNA, absolutely. This is looking good, 7th period. Right? And what do we call that portion of DNA that codes for a protein? Go to the gene. Gene, very good, okay? So it is my genes that dictate primary level of structure, dictate the number and pattern of amino acids that make up that particular protein, right?
So the genes dictate
The gene has the instructions, right? The original copy of the instructions. As we know, in the actual process, we make a disposable copy of the instructions, so the gene has the original copy, yeah? Okay, so when we look at amino acid structure and think about what an amino acid looks like, we know that we have that amino group, and we know that we have that carboxyl group. Oh, by the way, I forgot to do the terminus, didn't I, on that diagram? I'm sorry, you guys should have stopped me. I got all excited because you do the functional groups. Terminus, what would the name imply that might be? The end, right? Proteins have endedness. Polypeptides have endedness. There's always going to be an amino end, which we call the amino end, the amino terminus, or the end terminus. And there's always going to be a carboxyl end. And we refer to that as the carboxyl end, the carboxyl terminus, or the C terminus. Just terminology that you may come across. Okay? All right. So back to primary level or secondary level of structure. So what do we remember about nitrogen? It is polar. It's buff, right? It's electronegative. And so we know that when we look at those hydrogens that are attached to that nitrogen, because nitrogen is pulling those electrons closer, what's going to be the slight charge on those hydrogens? Positive, correct? Likewise, when I look at that carboxyl group that makes up an amino acid, which of those partners is pulling electrons closer? The carbon or the oxygen?
Oxygen.
The oxygen, right? And so that oxygen is going to have a slight negative charge. Everybody agree? Okay? What do we know about slight positives and slight negatives? They attract, and specifically they hold hands, right? What do we call it? Hand holding. Hydrogen bonding. So what happens with this chain of amino acids is we have that attraction between the hydrogen of the amino of one amino acid and the oxygen from the carboxyl of another. They're attracted to each other. They have a stickiness. They hydrogen bond. That results in two different conformations. One we describe as an alpha helix. It's a coil in which we have this hydrogen bonding that's occurring between that hydrogen of the amino group and the oxygen of the carboxyl group, okay? An alpha helix. Sorry, let me put it up here.
Those are the hydrogen bonds? Okay.
Hydrogen bond, yeah. Hydrogen bond is forming between the hydrogen of an amino group and the oxygen of a carboxyl, okay? Because of the electronegativity of nitrogen and oxygen, okay? All right, so this is what this looks like. It's called an alpha helix, okay? It's a coil. But we can also have regions in which we have hydrogen bonds occurring between parallel parts of the strand, and we refer to that as a beta-pleated sheet. It's the same thing happening, a hydrogen bond between the hydrogen of an amino group and the oxygen of a carboxyl, but it just has a different configuration, okay? And the reason for that is because of the R groups, but kind of outside of the scope of this class. Now, what's important to understand is within the same protein, we can have areas in which we have beta-pleated sheet and areas in which we have alpha helix. In other words, it doesn't mean the entire polypeptide chain is in a coil, okay, or in those parallel strands. And this, in part, gives some of the properties that we associate with proteins. Alpha helices are associated with a lot of our fibrous proteins, things like collagen. Beta-pleated sheets, we see in some of our stronger proteins, things like silk, okay, have a lot of beta-pleated sheets holding that conformation in place. But the key to remember here is secondary is hydrogen bonding, okay? Associated with hydrogen bonding in the polypeptide backbone. When I talk about the polypeptide backbone, I'm not talking about the R groups. Okay, I'm talking about everything but. Okay? All right, the next level of structure is tertiary. And this is, I would say, the most important part of the overall 3D conformation of that particular protein. What I'm highlighting in my notes here, making sure I write down, tertiary involves all types of interactions between R groups. And this is where the property of those R groups becomes so important, because that dictates what types of interactions can occur between them, right? So what types of interactions? All the ones you've learned about in chemistry. Everything from hydrogen bonds to van der Waals forces to ionic bonds to covalent bonds. All of those can form between those R groups and cause this protein to have its specific shape, its specific conformation that it's supposed to have. So let's take a look at this a little bit more closely. If I've got hydrogen bonding occurring between those R groups, what can you tell me about the properties of those R groups? They are polar, right? They are polar R groups if we've got hydrogen bonding occurring between them. Does everybody agree? What if I've got ionic bonds occurring, forming between R groups? Charged. We've got charged R groups, right? That's where we're going to see those ionic bonds form. Okay, van der Waals forces. This is a simplified definition, but it works for our purposes. Van der Waals forces are basically hydrogen bonds between nonpolars. Okay, even nonpolars, those electrons, because they're orbiting randomly, can result in slight charges. And so van der Waals are stickiness between nonpolars. So those are going to occur, obviously, between our nonpolar R groups. Now notice disulfide bridge. What amino acids do we say are always involved in a disulfide bridge? Sulfhydryl functional group, which is only found in cysteine. The sooner you learn that, the better. Okay, so that's going to be a very strong covalent bond that forms between two cysteines, and that is critically important to protein function and protein conformation. You have visible evidence of the quantity of cysteines that you have in your hair, because if your hair is naturally wavy or curly, it's because you have a much greater number of cysteines, and those disulfide bridges cause the hair to kink a little bit. Whereas if your hair is straight, you don't have as many cysteines, you don't have those disulfide bridges, and so your hair hangs straighter. And as those of you know that have curly hair, you can put on a straightener, a chemical that breaks those disulfide bridges, but ultimately they reform. Ultimately they do come back as a result of that. And we'll look at some more evidences of the importance of cysteine a little bit later. But that tertiary level of structure, what am I associating with that? What word did I tell you to highlight? R groups, okay? That's where we are talking about interactions between R groups. And again, the properties of those R groups so important. If I have a protein in solution, okay, what does it mean if it's in solution? It's in water. So when that protein folds into its conformation, what would you predict about the R groups, the amino acids that are on the outside of that protein facing water?
Hydrophilic.
They're going to be either polar or
Hydrophilic.
charged. They're going to be hydrophilic, right? Whereas the inside of that protein, away from the water, because that's what causes this protein to fold, right? What types of amino acids am I going to see in the inside of this protein? Nonpolar, okay? So it's really important to understand the properties of those R groups and how that is going to relate to the way that protein comes to its normal shape, its conformation. So what's left for quaternary? Well, again, remember, not all proteins have quaternary level of structure. In order for a protein to have quaternary level of structure, it has to be composed of more than one polypeptide chain. And quaternary level of structure is simply all those interactions we've talked about, but it's occurring between two separate polypeptide chains, okay? There are some good examples of proteins that have quaternary level of structure, are composed of more than one polypeptide chain. At a molecular level, how could that happen? How could I have a protein made up of more than one polypeptide chain? How would that happen? What would that tell you about the instructions for that protein? found in more than one gene, right? So for hemoglobin, we have two genes that code for hemoglobin A and hemoglobin B that dictate how our hemoglobin molecule basically is formed, all right? Another way that can happen is if we have a polypeptide chain that's created, synthesized, and then it's cleaved in half and then allowed to interact. But it means there's more than one polypeptide chain. Okay? Any questions on that, guys? Is that a yes or no?
No.
All right, you're sitting up straight, actively listening. I can't even tell you how important this material is. Strongly encourage you to read about it in your textbook, listen to a devotion video, or something to reinforce it. Teach it to me. All right, we're going to look at this tomorrow. So how do we ensure that protein folds properly? We've already talked about some diseases in which we have improper folding of proteins. For example, Alzheimer's disease, those prions that we talked about, right? Abnormally folded proteins. Proteins that are folded abnormally cannot do their job. And we have those proteins, so they do a specific job. So that always is going to cause us problems. So what we have discovered is that we have structures in our cells, as do all living organisms, including even bacteria, called chaperones. And they've been given that name because their job is much like a chaperone. What does a chaperone ensure? Yeah, watches over, exactly. Make sure things go the way they're supposed to. So what chaperones do is they provide an environment for proper folding of that protein within the cell. What might be important? Well, for example, the pH, right? Make sure the pH is appropriate so that protein folds properly. So these are found in all cells. We didn't even know they existed 10 years ago, and now we recognize that all cells have them, and they are critically important to the conformation of that protein, to ensuring that it folds properly. It's threaded in its primary structure, and it's allowed to fold into its normal conformation within that chaperone. Okay. So why do we need proteins? Well, there's a vast number of reasons that you and I have to have proteins. These are some of them. We'll be talking about enzymes in just a moment. Our antibodies are composed of protein. I thought we talked about, that was in sixth grade. We use proteins for transport. Remember those protein channels that are embedded in the cell membrane that allow for movement of certain materials in and out of the cell. Most of our hormones are proteins. We've got receptor proteins on our cells that allow certain things to be received by the cell. You can read through this. But what I do want to mention to you is the importance of protein and protein conformation in cell-to-cell communication. What did Bonnie Bassler talk about? What that cell-to-cell communication between bacteria? Quorum sensing, okay? We also have cells that communicate with each other. That is based on protein conformation. In other words, hormones are a means of communication we have in our cells. Hormones are released into the circulatory system. How do they act upon a certain cell? Because of the fact that that hormone has a certain conformation and that cell has a receptor protein with a certain conformation, and they are compatible with each other. It's the same principle with quorum sensing, right? Those bacteria send out signaling molecules. The bacteria that receive those molecules and react to them have a protein receptor that has a comparable conformation to the signaling molecule itself. So conformation, critically important. Alrighty, let's switch gears and talk just a little bit about enzymes, kind of lead us up to the lab that we're going to be doing tomorrow and help us to understand that. So you remember learning about enzymes in Bio 1, yes? What is the job of an enzyme? Okay, it allows reactions to proceed much more quickly. Are these reactions that would normally occur, or do they occur because of the enzyme?
Normally occur.
These are reactions that would normally occur. Okay, so what enzymes do is they catalyze, they allow reactions to occur much more quickly. Those are reactions that would normally occur, but it speeds those reactions up. You and I cannot live without enzymes. We have enzymes in our body that are constantly triggering chemical reactions and allowing them to proceed at a rate that is compatible with life. So how do they do that? Well, as a reminder, we're going to be talking about this much more in 2 units. As a reminder, every reaction needs a little push to get it going, right? And that push is called activation energy. And so what an enzyme does is it decreases the amount of push that is needed to get it going, right? Decreases the activation energy. So, here is an illustration to show that. Here is the reaction occurring, what it takes without the enzyme. This shows the reaction with the enzyme. Important to note that the change in energy, the delta G, we will be doing delta G, you probably love that, yes? Yes. Delta G is the same with or without enzyme, it's just that push to get that reaction going that's different. So, your question should be, how does it decrease the amount of push, right? What's the answer to that? Nobody knows? Good thing you took bio too, yeah? Good thing I happen to have an enzyme here. All right, so here is my enzyme, okay? As you recall, this enzyme will only catalyze a specific reaction, right? Right?
Yeah.
Okay, good, make me feel better. All right, what do we call this substance that is gonna be acted upon with the help of the enzyme? That's the substrate, right? Enzymes are substrate specific. Each enzyme is meant to catalyze a specific reaction, yeah? How do we ensure that specificity? What do we call the region of the enzyme in which that substrate fits? The active site, right? So we have that active site that is compatible with the substrate. Now, if you learned that this was like a lock and key, I want you to banish that thought from your head forever. And don't ever say that and then say the word bio too, okay? Because it's not a lock and key at all. It is not something that is rigid in any way. Rather, instead, when that substrate settles into the active site, we have a situation we call induced fit. And this principle is so important, I'm 99.99999% certain you're gonna have to explain this to me on your FRQ, okay? What is the importance of induced fit? So I'm gonna tell you. The way that I think of induced fit, because I'm a little bit of a romantic, is it's like a hug. When that substrate settles into that active site, there is a hug that takes place. There is kind of a settling. That enzyme kind of just molds into that substrate. It's not a perfect fit to begin with. It is specific. Only a certain substrate will fit in that active site, but there is an enveloping, a slight change that occurs. Like I said, I equate it to a hug. It is the hug that lowers activation energy. What? How does that happen? Well, let's say that this enzyme is catalyzing a catabolic reaction. Is a catabolic reaction building or breaking? It's breaking, okay? So how would that happen? When that substrate settles into the active site and that hug takes place, it stresses the bonds holding that substrate together to make it more easy for those bonds to be broken, okay? On the other hand, what if this is an anabolic reaction? That substrate settles into the active site of that enzyme. What does that hug do? It holds the components very close together, so it's easier for the bonds to form. Either way, that hug, that induced fit, is what lowers the activation energy. Amazing. Yes?
This is probably a dumb question.
No, I'm
but-
sure it's not.
But if it squeezes both of them, how does it make one form bonds and the other one, like, break bonds?
Because in the case of breaking bonds, the bonds are already there, so that squeeze
Okay.
is gonna put stress on the bonds that are already there.
So the other one is like two separate things.
And so the other one is two separate things, it's holding them together so those bonds can form.
Okay.
Okay? Yes?
And, I forgot what.
Okay, well, if you think of it, you can ask.
So the substrate goes to the enzymes?
The substrate fits into the active site of the enzyme. This whole thing is the enzyme, this is the active site. Right.
How do they get out of it?
How do they get out of it?
Yeah.
And that's an important principle, and it's really important with our lab, right? So how do substrate and enzyme find each other? Well, remember, what did we say about molecules and atoms? What are they always doing?
Moving.
They're always moving. So it's just that random movement, those random collisions, and the fact that they continue moving once that reaction's occurred. Okay?
How is it considered lowering energy? Is it just lowering energy for the substrate to do for itself?
It's lowering the push needed to get the reaction going. That's what we mean by lowering the activation energy. It's just making it more, it's making it easier for the reaction to occur. Remember, this is a reaction that would normally happen anyway. It's just facilitating that reaction, making it easier for it to occur. Any other questions, guys? Okay, all right, we're gonna go back to the back. I believe I've given you 15 minutes. Guys, when you get to the back, one side of